Quick. There we go. Okay. So this is a short summary of our HFSP project. We are interested in a quite general phenomenon, namely that many insects eat plants, and they eat them quite a lot. So about 20% of all calories meant for human consumptions are still consumed by insects. And the perspective that we took on this was, well, if you want to eat, you have to mechanically process the food, much like you do when you chew, and that should take mechanical force. And that mechanical force depends on how much you have to apply on something like the material properties of the material that you're feeding on. Could be very soft, could be very hard to eat. And the tool that you're using to feed, sharp teeth versus dull teeth. And to produce that force, you need to use muscle, and that costs metabolic energy. So it's a process that is mechanically costly and metabolically intense. And our simple idea was, well, if that's true, then these two together should interact with animal size, with what the insects feed on, to determine something about how these animals actually behave and how they can be efficient in feeding on plants. So that was the general idea, and that means that initially we were interested in things such as how quickly insects can feed on plants, or how long it takes them to cut certain fragments of plants. And if you could play the video for me, please. There's one example of an individual that we're interested in, leafcutter ants. And you can see here that ant cutting fragments from a leaf, and you can see that it's really digging into that leaf. It's a physically very intense process. And we were basically interested in a simple question, well, how long does it take the ant to cut a fragment of a certain size, depending on the size of the ant, depending on the plant material that it's cutting, etc. So this is a mechanical problem. Now, I'm not sure whether it was on for long enough, but we got our Young Investigator Award in 2020, so timed it perfectly almost with COVID. And that resulted in quite a lot of problems initially with our experimental progress. So when we started thinking about this, we were initially restrained to purely theoretical considerations. And then we very quickly realized that, of course, muscle to move stuff is used not only by small ants or insects in general to feed on stuff, but in all sorts of contexts and scenarios. We use them for very nice things. We use them for sometimes not so nice things. They're used by very large animals, and they're used by very small animals. They're used by animals that fly, that swim, or that walk, so in all sorts of different scenarios and contexts. And despite that extreme diversity, muscle as a tissue and the molecular mechanism of muscle contraction is highly conserved. So really, all these muscles work in plus minus the same way. And if that's true, then our idea was, well, if we want to think about what you can do with muscle, and there should be some shared constraints that apply not only to these tiny insects feeding on plants, but more generally to animals across all size scales. So I'm going to ask a very naive question, and I'm going to allow myself to simplify a musculoskeletal system as this very simple sketch here. You have just some mass, that could be my arm, or that could be the body mass, and you have a muscle here. And you want to know, well, how fast can that muscle make that mass move? It's a very simple question, and as a physicist certainly you would say, well, that's a question for the conservation of energy. If you want to make that mass move, you have to accelerate it, you give it some kinetic energy. And how much kinetic energy you can give it depends on the amount of work that you do. That's a very simple physical statement. And indeed, that's how people have thought about this problem for a long, long time. So probably going back all the way to Borelli, a contemporary of Galileo. Galileo developed mechanics. Borelli was like, well, I can use that to study movement in humans as well. And then you could say, well, that's quite a simple problem. You have a muscle of a certain volume, can produce a certain stress, and can strain or shorten by a certain amount. And the product between these two gives you the work. And then you say, well, you can move that mass as fast as you can do work, and that depends on these parameters. And that's what in biomechanics research is typically called the Borelli limit, and it has been around for a couple of hundred years. That's nice and simple. But when you think about this a bit more deeply and think about muscle physiology, then it turns out that that's not the whole story, because there's a second limit that is imposed by muscle. And that is that muscle also can only shorten with a maximum strain rate. It can't shorten arbitrarily fast. So muscle also has a maximum velocity with which it can contract. And if there's a maximum velocity, that means that because this mass is coupled to the muscle, that mass can never move more quickly in this simple system than this muscle is contracting. These speeds must be equal. And if these speeds are equal, then it must mean that there's a second limit to how much energy you can inject, and that is that not the left side of this equation is bound, but the right side is bound. So there's a limit on the kinetic energy capacity. You cannot inject more energy than the square of the velocity of this muscle multiplied with the mass. So there are two fundamental limits then for how quickly muscle can make things move. Okay, there's two. Well, which of these applies? And now we have two physical quantities of the same dimension. They're both energies. And what physicists like to do then is, well, we divide one by one another, and then we get a dimensionless number. There's no longer any physics associated with that number. It's all here. There's no units left here. Now, you can write that number in terms of physical parameters alone, and you can write it in terms of the product of three terms. One has to do with the physiology of the muscle. so it's something like a stress, a strain rate, and a strain. One has to do with how long that muscle is for a given volume. So you take a bit of volume of muscle and you either allocate it to area or to length. And the last one tells you how much motor, if you will, you invest to move that mass. So it's the density, how much muscle you have per size. Okay, you can do all these, and then you can create relatively simple plots where you can say, as a function of this one number, how much energy can that muscle actually inject? And you can split it in two halves. If that number is smaller than 1, then that muscle is limited by how quickly it can contract. And if the number is larger than 1, then that muscle is limited by how much work it can actually do. So it's a relatively simple plot, and that means if you're left of this number, you can't actually do all that work that Borelli and his fellows would tell you, but you're restricted to a smaller amount of work. And then as a biologist, the intuitive question is, well, what value of this parameter gamma does muscle actually operate with? And the nice thing is, because these are physical parameters that you can measure, you can go to animals or you can do some anatomy, you can do some physiology and measure all these, you can calculate that number. You can put values to it. And here are just a couple of illustrative examples from a shrew to an elephant. And the point I want to get across here is that that number depends on animal size. And that size dependence arises from this muscle length in here, and that means that larger animals typically have longer muscles, and that means they have a larger value of this number. And that means if you're really small, you're limited by different things than if you're really large in terms of how much energy your muscle can output. So that's a prediction, and that prediction has some consequences, and I'm going to briefly illustrate one before I go to talk a bit more about what you can do to cheat with these limits. So this is old data collected by Ted Garland in the 1980s, and it's plotting the maximum speed with which animals can run against the size of animals. And the finding is relatively intuitive. Larger animals are generally faster. A cheetah is faster than a mouse. But the fastest animals are not the largest. Elephants are not faster than a cheetah. So speed first increases with animal size, and then it flattens out. And this observation has been around for a while, and basically every 4 years there's a suggestion for what might explain this, because this is quite an unusual relationship in biology, where we normally expect that things change continuously with size. My head size goes up when I get larger for animals. Heartbeat goes down as animals get larger, right? So normally these relationships are continuous, but here it's not. It's a discontinuous relationship that changes. Now, if you look at this relationship from the simple perspective of this very simple physical argument that I've just made, then we know that if you are below the certain dimensionless number here, you only have access to some of your work capacity. And if you're above it, then you have access to all of your work capacity. And we know that this number varies with size, so we know that the amount of energy animals can inject should vary with size up to a certain point. And we know that we can directly calculate that number here, so we can exactly calculate when animals should no longer be able to get faster, and up to which size they should be able to get faster. And this is what you get when you do this. It's a direct calculation. There's no fitting involved. You just simply plug in some physical parameters into that number, and you predict that the fastest animals should be about 50 kilograms, about the size of a cheetah. And that is because they're exactly at that sweet spot where they can deliver optimal amount of energy, and above that you have access to about the same amount of energy. Okay, great. So this is just a simple example of what you can do with a relatively simple physical argument like this. But as a biologist, you might want to take a step back and say, well, this is also a bit weird, because we normally think that evolution would, through selective pressures, ensure that systems have some form of design that allows them to perform in a reasonably good way. And if you have a design where you're a small animal and you sit somewhere all the way back here, then you only have access to a tiny capacity of your muscle. Right? So it seems like you're over-designed. Why would you waste all that design? So what I want to do for the last 5 minutes or so of this talk is talk a little bit about things that you can do to cheat with this physical constraints, so how you can have musculoskeletal design that allows you to overcome these limitations. Now, I only have time to talk about one feature, but the feature I'm going to talk about is that muscle very rarely attaches directly to the mass that it moves, but it attaches to the mass that it moves via tendons, or apodemes in small animals. So they're elastic animals, so you can think of this as something like a spring. Right? It stretches as the muscle applies a force, and we're going to give that spring a certain spring constant, K. Now, if we go back to this simple energy balance, what that means is that as muscle does work, that work doesn't only increase the kinetic energy of that mass, it also introduces some stored strain energy in that system. Right? It stretches that spring, and then that spring, it stores some strain energy. Now, if you look at this from the original perspective of saying, well, muscle is limited by its work capacity, then this shouldn't really change very much because it always does the same amount of work W, and you just change whether that work goes first into strain energy and then in kinetic energy or not, but you do the same amount of work. So from that perspective, springs are completely irrelevant and your tendons do not have a meaningful impact on what you can do with your musculoskeletal system. But if you come from this other perspective, when you say that muscle really is limited by this kinetic energy capacity, then it's a bit unclear how much work it can actually do because it has to get to this kinetic energy capacity, and in doing so has to stretch that spring. So maybe here, adding that spring and giving it the right values allows you to overcome this constraint that I just introduced. You could do this by writing down the equations, but I don't think that would be very helpful, so I'm going to try and talk you through this with an intuitive example where I look at extremes. So I'm going to go back to this very simple equation. It tells us that the work that is done is equal to the change in strain energy, stretching the spring, and the kinetic energy, accelerating that mass. And I'm first going to talk about an extreme case, and that is that we're going to make that spring very, very, very stiff. If that spring is infinitely stiff, it shouldn't make a difference that it's there. The system should behave exactly as before. And what happens in that case is that you store some strain energy that goes inversely with that spring constant, and you do roughly the same amount of work as if that spring wasn't there. You deliver roughly your maximum kinetic energy capacity. That means that the ratio between these two energies is much larger than 1, and with that you can calculate what a very stiff spring should look like in terms of the system parameters. At the other end of the extreme, I can make that spring very, 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 very floppy, so it's very easy to stretch. Now, if that spring is very floppy and that muscle shortens and contracts, this mass will hardly move, and all I will do is stretch that spring. Right? So by the end of that muscle shortening, the spring will be stretched, but the mass will pretty much exactly be where it was before. So in that case, the energy that you can store in the spring is limited by how much that muscle can shorten, and you should basically just inject this maximum amount of energy in the system because you can't really accelerate the mass. The force it experiences remains slow. In that case, again, you have a ratio of two energies that is much larger in unity, and you can find a limit for that spring constant that tells you how soft is soft. Right? It becomes quantitative. Now, the third case that gets really interesting is, well, what happens if your spring is neither flippy or stiff, if it is of some intermediate stiffness? What happens in that case is this muscle shortens. As it shortens, it stretches this spring, but it also accelerates the mass. And it can stretch the spring exactly until the point where this mass is moving with the same speed as this muscle is shortening. Right? At this point, this end of the spring and this end of the spring are moving with the same speed. The spring no longer stretches. And at that point, the energy in the spring must be exactly equal to the kinetic energy capacity of the muscle. So you do more work than you can do without that spring. And you can say that that spring without, or in general, has to fall between these two extremes of a very stiff or a very soft spring. Now, you can calculate this all exactly, and you can plot something like the energy output that your muscle can produce compared to the stiffness of that spring. And this is what the muscle could do without that spring. And you can see that if you tune your system nicely, so if you give your tendons just the right stiffness, then you can perform much better than you could do without that tendon. Now, that's a direct prediction, because again, this tells us something about the stiffness of tendons, and we can go out and measure the stiffness of tendons and see how well they do with, compared to the theory. And here's some data taken from the literature for tendons of animals across a range of different sizes. And this dashed line here is a direct prediction, again, from the physical parameters. Not fitted, this is measured and then predicted with that simple mechanical argument. And it's relatively, arguably, relatively close to what stiffness these tendons actually have in real animals. Now, with that, I'm practically at the end of it. So what I want to say with this talk is that sometimes research takes unexpected turns, and we started with this relatively naive question about how long does it take that ant to cut that bit of leaf. And we did also answer that question. I didn't talk about it, but we can apply the same theory to that question. But you can then also take these things sometimes further. And it looks like that from simple dimensional arguments like the one I showed, muscle hasn't just one energy capacity, like what was thought for a long time in biomechanics, but two. And it looks like the second energy capacity that has to do with how quickly that muscle can shorten is much more important than the first. And if you take these two arguments together, then you have a tool with which you can try and explain some observations about how animal performance, maximum speed, endurance, etc., changes with animal size. but also how you can think about design of musculoskeletal systems. What are the physical parameters that you want to give your muscles and your tendons to achieve optimal performance? With that, I would like to thank my research group, my collaborators, and of course the HFSP for funding fundamental science. You're very much for your attention, and thank you very much.
Yeah, thank you very much. Also for reminding us that size matters in biology, which I like as being a microbiologist. First question, please. Yes, please.
Thanks for the really interesting talk. I was wondering, in your plot that you just showed of the stiffness versus the body mass, it seemed like the points were all a little bit below the curve, so they're less stiff than they should be according to the model. What are your thoughts on why that might be the case?
Yeah, I mean, there is lots of uncertainties also in these parameter estimates, right? So we have to measure the tendon stiffness, the amount of muscle, how that muscle is split in the stress. So I think that's probably observed somewhere in the error propagation. So if I would plot that with error bands, it would look more sensible, and that would be my best guess towards that. Now, it's a relatively simple estimate, and it's within a factor of 2. So for physicists on that scaling plot, we're quite happy with that. From a biological perspective, there's open questions, and we're trying to build simple model systems with which we can test some of these predictions more directly too. Thank you.
Thanks.
Yeah, thanks for the interesting talk, and I guess I'll be asking a question that maybe comes from a very different perspective, but in biochemistry, for instance, when you think about the free energy that is released when ATP is hydrolyzed to, say, promote a muscle contraction, some of that would go to also increasing the entropy of the system. Not all of it would be directly coupled into mechanical work. I'm just curious if that's a parameter that you think is interesting, that's different from organism to organism, or can it just be treated as a sort of an offset that just applies universally that doesn't really affect the sort of macro biomechanics?
Yeah, so we very shamefully ignored that in this very mechanical perspective, but it's a very interesting question because it pertains directly to the efficiency of locomotion. And there seems to be data that suggests that larger animals are more efficient, and it remains relatively unclear as to why that exactly is the case. There are some hypotheses. Some have to do with the use of tendons, as I just showed. But we're very interested in that and now trying to link that back into that purely mechanical perspective by also thinking about the thermodynamics of the problem. Thank you.
Exactly. We would have time for another question. Okay.
Thank you for the interesting talk. I was wondering if viscosity might challenge these limits due to elasticity, so the relaxation time of the tendon. Does it introduce some sort of variability to the theory?
Yeah, so it doesn't really change the fundamental limit. That remains actually the amplification factor that you get remains the same. It changes a little bit what happens left and right away from the optimal stiffness.
Jörg, there's one more here eventually.
Yeah, we'll take another one. Take the opportunity.
Thank you, fascinating talk. So I was just wondering, so you are taking it as a standard, so that muscle at the standard level, but then this differs during age. So how does that differ for the different sizes and velocity with age? Is it proportional or is it very different?
Yeah, so definitely age has a huge influence, in particular on stiffness, and also stiffness between muscles in the tissue. So I think this sort of very broad-scale argument works well if you look across decades of size. If you look within one specific group or within, say, humans, then you have to do much more accurate modeling and measurements for sure. In general, the assumption would be that if muscle could perform much better, then why wouldn't it evolve across all sizes? Right, that's the general assumption, and the data that exists suggests indeed that the variation of physiological properties of muscle is relatively small. So across the 10 orders of magnitude of mass in animals, they vary by at most an order of magnitude in size, something like stress and strain rate, and there's no systematic trend in that data.
Okay, thank you again.
Thank you.